Hi everyone, hope you're doing well. I thought I would just make a quick little video for you guys on the visual pathways because I understand you're not getting any teaching just now with everything that's going on with the coronavirus, and obviously head and neck's quite a heavy section to go through, so I thought I'll try and help with this because a few people had mentioned that they were struggling with visual pathways, so I hope this helps. Just a wee disclaimer, I'm really prone to getting tongue-tied, and there you go, tongue-tied, and a bit of a stutter, so bear with me if I struggle to get words out a wee bit. So anyway, let's get started. So here we are, we've got the brain in front of us, and we're looking down on it from this angle. I've drawn it so we're looking down on it, and we've got our eyeballs here at the front, okay? And this blue line is representing all of your left visual pathways, okay? And what's important to understand is that these blue lines are representing your right-sided vision, okay? So this is your right side, and the left retinas, both in the left eyeball and in the right eyeball, are taking in information this way, okay? So your left side is processing your right side of vision, and it comes straight away. As soon as it hits the retina, it's your left retina that's processing your right side of vision, and that gets transferred onto the left side of your brain, and your left side of your brain processes that information. And that's very important to know because it means that if you have vision loss on one side for whatever reason, you can narrow it down to know that the pathology is somewhere on the left side. Okay, so let's get to grips with a little bit of anatomy just so we're aware. So this here, this portion here, where we circle, this is your optic nerve. So this is your optic nerve. This is also your optic nerve, and it is combined together with the right side of the retina, and it passes through, the optic chiasm. Another disclaimer, my spelling is absolutely atrocious, especially when I'm writing fast. This here is your optic tract, so that's that, and these are your optic radiations. And important to know about the optic radiations is that they're very diverse. There's thousands of fibers and they're really messy. They look like this. I don't know if you've had a chance to see them in dissection in the anatomy labs, but they're crazy. The optic radiations, however, are kind of split into two main bundles: the temporal optic radiations and the parietal optic radiations. Okay. So just to go back through this, is that you've got your left retinas here, you've got your optic nerves which come down, and then the opposite side branches off and passes through the optic chiasm, and then the same side continues on and they join together here to form the optic tract. They then pass through, and they actually pass through something here which is important to know for visual processing, something called the lateral geniculate ganglion, and from there they spread into the optic radiations, there's two main bundles, the temporal and the parietal, and then they go down to your visual cortex to be processed and then relayed forward to your conscious mind so you can see. Okay, dokes. So the important things to be aware of is if there's disruption to any of these pathways, how will your vision change? And this is something that you get quizzed on. It came up in my second year exam, still comes up in exams. It's important to know. Okay, so first things first is that if you were to damage the optic nerve, so if you were to have damage here, cut this, what would you expect to happen? Well, remember you've got two sets of visual pathways sort of coming through here. Let me put this in red to help clarify it. You've got your left retinal information coming in, which is taking your right side, and then you've got your right retina, which is taking in information from the left, so the opposite side, and these fibers would pass through together as the optic nerve, and then the right retina would branch off and go pass through. Sorry, it's not quite equally drawn there, but would pass through the optic chiasm onto the opposite side. Now the point is that all your information from the left side and the right side is passing through your optic nerve. And as a result, if you damage your optic nerve, you can expect to wipe out all vision from that eye. So all the vision from that eye will be disrupted. If you have damage to your optic chiasm, okay, and this is mainly due to things such as pituitary tumors, such as prolactinomas and adenomas, what you're going to get is you're going to get damage to the outside of your vision. And you can see this because your right retina, which is taking information from the left side, i.e. your temporal side, will be disrupted, and same with your right eyeball, that your left retina, which is taking information from your right side or your right temporal, will be disrupted. And as a result, you're taking out information from here and here. So if you look at your eyes, you'll disrupt the vision here, and you'll disrupt the vision here. Okay, so I hope that makes sense. It's because what you're doing is you're taking out your peripheral vision on both sides. Okay, and this is called bitemporal how we spell hemianopia? Hemianopia. I think that's how you spell it. Anyway, bitemporal hemianopia. Okay, and it's all to do with the crossing over of the vision. So let's just clear this up a wee bit because it's a bit messy, and hopefully you can see it just a little bit better. I'm just going to rub these out. There we go. So your information coming in from this side and crossing over is taken out, and your information coming from this side and crossing over is taken out here, and as a result your peripheral vision is disrupted. And of course both sides are called temporal, so this is your left temporal and this is your right temporal, and so therefore it's both sides, which is bitemporal hemianopia, disruption of vision. Okay, now if you have damage to your optic tract, by the point that it comes down here, all your right-sided vision has now mixed together into your optic tract. So your left retina on your left eye and your left retina on your right eye has taken in all the information from the right side and has joined it together into the optic tract. And so if you disrupt vision from here, all your right side vision is going to go, because all the parts of your retina which supply the right side have been disrupted. And as a result, you'll lose complete vision on the opposite side that's disrupted. So in this case, because we've drawn it on the left optic tract, you lose vision on your right visual field. And it's the same. And this is called harmonious hemianopia. And because it's on the right side, you call it right harmonious hemianopia. Okay, so I hope that makes sense there as well. You've lost all the vision from the opposite side. Okay, so going into the optic tracts, it gets a little bit more complex than this, because this can get split up into quadrants now. And this is one of the reasons why I've drawn these quadrants around here. And let me explain why. If I draw an eyeball, that's not a very good eyeball. Draw an eyeball like this, and I'm drawing it from the side here, so we're looking at the side. So this is the top of the eye, this is the bottom, and this is the front, this is anterior. Okay. Not only have you got left and right retinal fibers, but you've also got superior and inferior retinal fibers. So I'll do it in a different color here just to... So your bottom retinal fibers are your temporal. They go through the temporal radiations, okay? And then the top ones are your parietal. They all form into the parietal. And as a result, because they're on top, they take in information from the inferior aspects. And vice versa, because the temporal is on the bottom, it takes in information from the superior vision, okay? So because the parietal fibers are on top, they take the inferior information, and because the temporal is on the bottom, they take in the superior visual information. And this means that if you lose vision in a particular quadrant, you can narrow it down. So let's explain this a little bit more. If you were to damage here the temporal optic radiation, okay, what you're doing is you're disrupting these fibers here, and so you're going to lose your superior vision. So what we know is we know that it's either going to be here or here that's disrupted, and because it's on the left, we know it's the right side. So we lose the upper right quadrant vision, and this would be in both, because remember the fibers cross over. Okay? So because the temporal fibers are on the bottom, receiving the superior visual information, your superior visual information is lost, and because it's on one side, it's just on one side. And here, for example, your parietal fibers are still working fine, so you're still receiving the lower visual information. But then, if you were to damage your parietal radiations, then what you would get is you would get a lower quadrant, so we know that it would be one of these two, and then of course it's on the left side, so it would be the right side information that would be destroyed, or not destroyed, but unable to be processed. So you'd get lower quadrant, right lower quadrant visual field defect. Okay. Now, moving into the visual cortex, the visual cortex is actually in an area, so this area here is actually called the Cal-Serena fissure. And there's two bits. I'm going to draw it just out here a wee bit, so if it's just to draw this. So this is me just zoomed into this area here, okay? And this is your grey matter that's sitting around here, okay? So this is your grey matter. Hope this makes sense. When you move slightly more into the fissure here, this area is known as the main visual cortex. This is the main visual cortex, and this area here is the macular processing area. Now the reason I'm saying this is because if you damage just this area of the brain, just this area here, you're just going to damage your macular vision. That's the only thing that's going to be damaged, and so what you actually end up with is you actually end up with vision loss like this. Okay, so if you see any sort of visual pattern loss like this, it can be due to the macular processing area being damaged, and of course that should be on the opposite side there. If it's, I know I've drawn it because I did it on the right side here, so their left side is correct. Okay, so I hope that makes a little bit more sense. I know it looks really messy just now, but the main things I would really learn would be your bitemporal hemianopia. That comes up, and the main thing to understand is that you can potentially just have a quadrant which is taken out, but if it's on both sides and on the temporal sides, it's always going to be the optic chiasm that's affected. Okay, and the reason I say that is because in my second year exam we had a visual field defect that just had the upper quadrants on both the temporal side that were affected, and everyone got a bit scared. But the point is that if it's on the both on the temporal side, it has to be where these cross over, and so it has to be the optic chiasm. Okay. If it's on the same side, so if it's harmonious and it's on the same side, then you know that it's across the optic tract somewhere, or both optic radiation or all the optic radiations have been taken out. Okay, And this one is quite clinically relevant because if you have a stroke, this is the most likely visual defect that you're going to get, and actually most likely going to be the right side as well, because it's usually the dominant side. And so as a case for this, the reason why is that it's the middle cerebral artery that's usually affected, and the middle cerebral artery supplies roughly all this area here with blood. And so if there's disruption to this, you're most likely going to get an harmonious hemianopia. Okay, I'm also going to very quickly touch on your visual pathway for your reflexes that you need to know about. So this is important as well. So when you shine a light in someone's eye, you get the pupil constriction, okay? And this occurs by traveling down through the optic nerve and then going down into the brainstem, and from the brainstem it gets relayed via the ocular nerve. And I'm going to describe all this, but just so you know. So when you shine a light directly into, say, the left eye and the pupil constricts, this is called the direct response. When you shine a light directly into the left eye, the right eye will also constrict, and this is called the consensual response. Again, I don't even know if that's how you spell it, but never mind. Okay, so why does it do this? Well, you need to know this pathway. What happens is light comes in through the left eye, okay? It will travel down through the optic nerve, and it will pass down through the optic tract, and it will go into this area called the lateral geniculate body, which we talked about. And from here, information will be sent further through your optic radiations for you to process that information and actually see. But there are fibers, I'm going to draw them in red. There are fibers that break off and go to the midbrain. And one of the places that they go is they go to the pretectal nucleus here within the midbrain, okay? And they synapse. And from here, they send fibers forward, and they also send fibers to the other side as well, to the opposite side of the pretectal nucleus. And that's why you get the consensual response as well, because it's here that the fibers cross over and send signals to both eyes, okay? So once the fibers have synapsed and sent across to the other pretectal nucleus, they then send fibers forward to a set of nuclei called the Egdinger-Westphal nucleus. And what this is it's an area of cells that are joined onto the oculomotor nucleus, but they are parasympathetic. Okay, they are parasympathetic fibers. And what they do, along with the oculomotor nerve, is go forward. And they go into the ciliary ganglion. So this is actually the preganglionic parasympathetic fiber. They then synapse in here, and then they go forward and go towards the iris, and then cause pupillary constriction. And this is an important pathway to know. Sometimes you can get tested on this. So it comes down, the light response comes down through the normal visual pathways, and when it gets to the lateral geniculate body, instead of being relayed on to your process of vision, it gets relayed on to the midbrain, and in the midbrain it goes to the pretectal nucleus, which then crosses the fibers over to the opposite side, so you get both a direct and consensual response. It then sends fibers forward to the Egdinger-Westphal nucleus, which is parasympathetic fibers, which is mixed with your oculomotor nuclei, which then projects fibers forward to the ciliary ganglion, and from the ciliary ganglion they go to the muscles to cause pupillary constriction. Okay, so I just thought I'd add that in there for just a wee bit more anatomy and the actual roots, and it's important to know this. And this is your midbrain that this occurs in, okay? And what I've done here is I've just drawn the superior colliculi, because that's where an anatomical landmark you should probably know. And this in here, this blue bit is your periaqual grey matter, which the aqueduct, so your cerebrospinal fluid flows through. Periaqual grey matter, you should probably just know for how morphine and opioids interact with this area to carry out their pharmacodynamic effects. Okay, hope that helps.